Our next speaker is Cynthia Wolberger, professor of biophysics and biophysical chemistry. Cynthia will talk about using one of our newest pieces of equipment, a cryogenic electron microscope, or Cryo EM, to understand how genes are turned on and off, and to apply that knowledge specifically to children with cancer who have a specific mutation.
All right, thank you very much. So will it let me start? I was getting messages. Will it let me start my video now?
Should be able to screen share, yeah.
All right, can you see it?
Yes.
Okay, great. Yeah, so thanks very much for the opportunity to talk about our work. So as James said, I'm going to talk about an exciting new approach that we have to look at the molecular machines inside our bodies. And I'd like to start with a story. So there's a type of leukemia called MLL rearranged leukemia, or MLL-r. It's a type of leukemia that is prevalent in primarily in pediatric patients, so really infants, or in patients that have been treated with certain types of chemotherapy, and it arises as a secondary cancer. Now, in contrast with many different types of leukemia, where there've really been terrific and highly effective treatments, this class of leukemia has just been resistant to any attempts to treat it. And it has a very poor prognosis, and we're still using drugs in the clinic that we used 50 years ago to treat this, so no new effective therapies. So our strategy in thinking about treating diseases like this is to really go back to the roots of human disease, to understand how the body works and how the disease causes it to malfunction. So as I see it, the challenge for the next 50 years in biology is understanding how the body works at the level of its component parts. So we have to know what the parts are, how they fit together, and how they work. And this is no different from understanding the machines that humans have made, like a car. So we know how a car works because we've designed it. And a car has something like on the order of 1,000 basic components that we've constructed, but humans are far more complex. We have something on the order of 25,000 basic components, most of which we've yet to understand. So just as you need to understand how each part of a car works in order to fix the car when it breaks down, we need to understand how each part of the body works in order to understand normal function as well as what goes wrong in disease. So here's an example of a car engine I found online. A car engine converts chemical energy into motion. And we know exactly how it works because we've designed and constructed it, but our bodies are also filled with equally, if not more amazing machines. But at this point, we really just know how a fraction of them work. So here's an example of one of these molecular machines that actually comes from the work in the lab of James Berger. So these are machines made of proteins, and that's what these cylinders here are depicting. So, most people, if you ask them about proteins, think of it as stuff that we eat. But in fact, the machines within our body are also made of these protein polymers that adopt different shapes. And those shapes determine what they actually do and how they function in our body. So next time you eat a steak, think about how you're eating all of these incredible machines. Now, these molecular machines are tiny. And I'd like to imagine, there's the machine that I showed you in the previous slide. And I'd like you to imagine that this molecular machine is the size of a car engine. So how big would that car be? Well, the car would actually be 145 times the diameter of the Earth. So that gives you an idea both of the tiny size of the machine as well as the challenge in studying it. Because how do we see such tiny molecular machines? We can't use a light microscope. We can't hold them still. And so to study them, we use techniques that use a different type of radiation. So either electrons in cryo-electron microscopy or X-rays in X-ray crystallography. And we're using both of these techniques, but I'm just going to focus today on cryo-electron microscopy. So this is not like the electron microscopy that Ken Whitworth talked about a little early, allowing you to envision a cell. But this is a new approach that's very excitingly just been developed in less than the last 10 years. There's been this leap in the technology that now allows us to use it to look at these tiniest of tiny machines in the cell. So I want to show you just a little video clip to give you an idea of the size of these instruments and to show you the instrument we actually have at Johns Hopkins. So hope this plays.
It's a warm summer day, but not everywhere. Somewhere deep underground, it's cold, really cold. How cold is it?
Approximately minus 180 degrees Celsius.
Wow, that is cold. I see wires, gold rings, and are those canisters of liquid nitrogen?
It's gonna be loud.
What is this place? What is going on here?
Welcome to the Cryo EM lab. So here we do cryo-electron microscopy, and basically we use a really fancy microscope to look at really small things, generally in the cell.
Why do you need to see these particles?
They are basically what make your cell run, and they're what make you, you.
How does this technology relate to the lab?
One of my colleagues here, Cynthia Wolberger, has used this type of instrumentation to visualize a particular enzyme. It's given her new insights into how we might develop small molecule therapeutics. This technology allows us, with much greater speed and accuracy, to be able to visualize how molecules give rise to life.
Okay, so that was a little tour of our Cryo EM facility in the basement. So I want to come back to the problem we were using Cryo EM to study. So I told you about MLL-r leukemia, an untreatable form of leukemia with a very poor prognosis. And a characteristic feature of these leukemias is that there's one molecular machine called DOT1L that is hyperactivated. It's misactivated in MLL rearranged leukemia, and as a consequence, it causes aberrant genetic program. So the wrong genes get activated and some of them hyperactivated, and that is somehow a part of the progression of this leukemia. So what DOT1L does is it modifies some of the proteins that package our genome. So we have, if you took all of our chromosomes and lay them end to end, there would be 2 meters of DNA, but they're not stretched out like that. They're highly packaged in the cell. They're organized by being wrapped around these discs of proteins called histones, and these in turn package tightly together and form the, during cell division, this sort of familiar form of the chromosome that many of you have seen. So we used cryo-electron microscopy to image DOT1L sitting on one of these discs of proteins. That the total disc is actually called the nucleosome. DOT1L is recruited onto the nucleosome by the attachment of one tag, the purple one over here called ubiquitin, and DOT1L in green then does its job of altering chemical tags that are on the proteins. And so I'll play this movie now. So what you're seeing here in gray is the actual electron density map that Cryo EM produces. Think of it as taking this entire molecular machine, the disk of proteins, the DNA, the DOT1L and these other proteins, and put shrink wrap around it. That's what that gray is, those outlines. And then we're able to interpret this map and construct a molecular model that fits it. So I've dissolved it away now and shown you the model we've been able to build of DOT1L. So we learned about how it toggles actually. This is DOT1L in green between these poised and active states. This is the disk, the nucleosome with DNA wrapped twice around it. This structure is the enormous wealth of molecular detail of what DOT1L looks like, how it moves around on this disk of proteins that's packaging our genome, and the details of the chemical reaction that it catalyzes in modifying actually this protein in blue over here. So one of the exciting things we learned about DOT1L that's relevant to disease is we learned that it has this pocket over here that's critically important. So over here in green, again, think if we've taken shrink wrap, put it around the surface of DOT1L, and what we discover that there's a pocket over here, and in this pocket, there's this bit of this other protein, it's called histone H4, that binds up in this pocket and turns out is critically important. Think of that red protein as a hand and DOT1L as the glove. If we pull this piece of protein away from DOT1L, then DOT1L no longer works. It doesn't do the chemistry it's supposed to on this histone. And so the idea is that taking this structural information, we are trying to identify drugs that plug up this pocket and prevent that bit of red protein from binding there, thereby inactivating DOT1L in a very targeted and specific way. And it's only because we have this structure that we've been able to determine with Cryo EM that we're able to come up with this idea. We didn't know about this before we determined this. Now, this is really just one example of an enzyme that modifies the proteins that package the human genome. And so I really want to end by zooming out and showing you the enormity of the problem. So here's DOT1L sitting on this disk of proteins surrounded by some of our genomic DNA. And again, that's just one very small bit of the packaging of our entire genome. So that back to this cartoon of the disk, and I'm zooming out to make the point that this is one very, very small part of what goes on in our cells in terms of regulating the packaging of DNA, which in turn regulates how genes are expressed, which ones are turned off and which ones are turned on. And there are thousands of these different protein machines, little pieces of protein that recognize these modifications on these proteins. And so there's really a huge amount to do and huge opportunities where we can exploit this technique of cryo-electron microscopy to learn about these very small molecular details and identify more Achilles heels in other molecular machines that we can target with drugs. So I just want to end by showing this is the team that have done this work, and particularly Evan Warden, shown over here, and Nick Hoffman. And these are the entities that have funded the research. And if you want to find out more about our work, you can check out our website. So thank you for your attention. Be happy to answer questions.
Great. Thank you very much, Cynthia. Just in the interest of time, we'll keep the questions brief. Can you just briefly touch on how you might use the structural information that you've gathered about DOT1L and that binding of the protein in the pocket to actually move toward something in the drug discovery side? How do you use that? What's the connection between that?
So there are a couple of different ways. Actually, one thing is you can actually take a structure and do something called in silico screening. If you know exactly what the pocket looks like that you want to target, there are already out there huge libraries of small molecules, and you can actually say, I want to target this pocket, and computationally search for the molecules that would fit in that pocket. And that gives you a starting point to test drugs and to design new drugs. Then the other way is we're actually designing specific screens that target exactly that pocket and not other parts of the protein. What I didn't mention is this enzyme is actually part of a large class of enzymes that catalyze similar chemistries. And targeting the sort of chemistry part of it, you're going to target every single other enzyme that does the same chemistry, but by identifying this pocket, we can design an assay for things that only bind in that pocket and do our drug screens in that targeted way. So those are two ways in which we could use this information.
Great, thank you. And actually, so there is one more question. So Peter Weiss asks, is Cryo EM a dynamic imaging method, or do you get static images that you have to imagine as a sequence?
That's an excellent question. So it's a static imaging method, but because of the way it's done, we actually look at millions of different particles of .1L at the same time on an EM slide. And so we can actually capture, imagine frames of a movie. We can actually capture something that's dynamic in all of its different positions, and then piece that together to arrive at the actual movie. And so that's what's exciting about that technique, and is something that X-ray crystallography, the other method I mentioned, is not very good at doing.
Wonderful. Thank you again.